This method can help answer key questions about Drosophila brain metabolism and how overproliferation of glial cells affects metabolic reprogramming, and whether diet or behavior alters metabolism. The main advantage of this technique is that fly brains can be metabolically analyzed as one whole intact tissue with reproducible results obtained from measuring just one brain at a time.
Though this method can provide insight into the metabolism of the fly brain, it can also be applied to other tissues and model systems such as imaginal discs and C. elegans, respectively.
To prepare the microtissue restraints, from the storage container, select 1 microtissue restraint per brain that is being measured, plus at least 3 for use as restraint-only controls. Using a mesh basket and a 6-well plate, rinse the restraints with 70% ethanol, and wash them with deionized water 3 times, each time for 2 minutes. Perform additional water washes if the restraints still give off an alcohol odor. Wash the microtissue restraints in assay media and leave them in this solution until they are ready for use. To dissect the Drosophila melanogaster larval brain, select the late third instar larvae from a vial of cultured Oregon R flies. Place the larva in the well of a clean dissecting spot plate containing 500 microliters of 1x PBS. Next, wash the larva by gently shaking it in the well, and transfer it to another clean well. Then, place the spot plate under the dissecting microscope.